Good day, this is Professor Will, CBMD PhD. Today, Saturday, January 11, 2020 at 6:37:28 p.m. Eastern Standard Time. And this is a reading on transcription elongation. So let's get started here. The initiation phase ends when RNA polymerase succeeds in extending the RNA chain beyond the first 9 nucleotides and escapes from the promoter. At that stage, the elongation process begins, and the enzyme starts moving along the DNA, extending the growing RNA chain. During the transition from initiation to elongation, the size and shape of the RNA polymerase undergoes successive changes. Whereas the holoenzyme covers approximately 75 BP from -55 to +20. After the loss of sigma, the polymerase covers approximately 55 BP from -35 to +20. At that stage, the polymerase is displaced from the promoter clearance or escape, and undergoes further transition to form the elongation complex, which covers only 35 to 40 BP depending on the stage during elongation. The polymerase now becomes tightly bound to both the nascent transcript and the DNA template, making it very stable. The average rate of transcript elongation by various RNA polymerases is 40 NT divided by S. However, the rate varies dramatically among RNA polymerase and loosely correlates with the subunit complexity of the enzyme. Thus, the simple single subunit bacteriophage RNA polymerases are the most rapid of all DNA-dependent RNA polymerases. Several hundred NT divided by S bacteria RNA polymerases transcribe at an intermediate rate, 50 to 100 NT divided by S, and eukaryotic polymerases, although diversified, appear to be the slowest, 20 to 30 NT divided by S. RNA polymerases are not as accurate as the DNA polymerase. The reason for the difference in fidelity between RNA and DNA polymerases might be that RNA polymerases do not have the robust proofreading mechanism that characterizes DNA polymerases. Of course, the fidelity of DNA replication is of greater importance than that of transcription because, unlike replication errors, misincorporation during transcription does not result in permanent and inherited genetic changes. Transcript elongation does not occur at a constant rate. Throughout the elongation phase, RNA polymerase can be paused, arrested, or terminated. During a transcriptional pause, the polymerase temporarily stops RNA synthesis for a certain amount of time, after which it can resume the elongation process. Thus, pausing can be described as a transcriptional hesitation. In contrast, during a transcriptional arrest, the polymerase stops RNA synthesis and cannot resume it without the aid of accessory protein. Throughout both pauses and arrests, RNA polymerase remains stably bound to the DNA template and to the nascent transcript. These features distinguish pause and arrest polymerases from those that have terminated and thus detached from the DNA. Pausing and termination are sometimes related because pausing is a prerequisite for termination, at least in the case of P-dependent termination. However, not all pause are termination precursors. The time it takes a stalled polymerase to resume elongation varies among pause sites from very short periods of time, which cannot be accurately measured, to several minutes. The fraction of polymerase that respond to an elongation block is also variable because ternary complexes differ in their ability to recognize pausing signals. Transcriptional pause and arrest signals can be intrinsic, that is, the sequences in the nascent transcript or the DNA template whose interaction with RNA polymerase can inhibit the progression of ternary complexes such as RNA regions, which have the propensity to form a stable secondary structure. In addition, extrinsic factors may obstruct the progress of RNA polymerase during transcript elongation. There are numerous examples of RNA polymerase from various organisms being physically blocked by DNA-binding proteins during RNA synthesis in natural and artificial systems. An example is the purine repressor, which binds well downstream from the pure B operant transcriptional start point and blocks the polymerase during elongation. It should be noted that in many cases RNA polymerase is able to transcribe beyond the DNA binding proteins in its path by either displacing them or bypassing them. In addition to DNA binding proteins, which are the most obvious obstacles for RNA polymerases, factors that perturb the structure of DNA can also inhibit the progression of RNA polymerase and thus interfere with transcript elongation. For instance, extreme positive or negative supercoiling, unusual DNA structures such as zDNA and DNA lesions. The efficiency of such potential impediments to block RNA polymerase from elongating depends on various local factors and on the type of RNA polymerase. For example, T7 RNA polymerase can efficiently bypass gaps in the DNA template strand that are 1 to 5 NT and less efficiently gaps as large as 24 NT. Transcriptional pausing is involved in various regulatory mechanisms. An example is the attenuation of amino acid biosynthetic operands, which depend on transcriptional pause that leads to the precise coordination of the position of RNA polymerase with the ribosome translating the nascent RNA. Although transcriptional arrest has been characterized in vitro and the evidence for its occurrence in the cell is only circumstantial, it is also believed to be implicated in the regulation of many genes. These predictions are based on the recognition that if an arrest occurs within the coding region of a gene, the arrest complex would block subsequently initiated RNA polymerases, thereby effectively repressing RNA synthesis from the affected gene. RNA polymerase and ternary complexes are able to endonucleolytically cleave the nascent transcript near the third transpose end. The resulting 5' fragment remains stably bound to the RNA polymerase, whereas the 3' fragment is released from the enzyme. Transcript cleavage does not result in transcript termination, but rather in the creation of a new third transpose terminus for chain elongation. Following the cleavage, RNA polymerase can correctly resynthesize the discarded RNA segment and continue with the elongation. The size of the released 3' fragment varies. There are reports of cleavage, products ranging from 1 to 17 NT. The cleavage rate also varies depending on the particular ternary complex. The mechanisms that cause the variation in both cleavage product size and cleavage rate are poorly understood. It is likely that the conformational changes that occur within the ternary complex as it moves along the DNA template play a crucial role in determining the rate of the cleavage reaction and the size of the cleaved fragment. The actual cleavage is carried out by the catalytic site of RNA polymerase, which catalyzes polymerization. However, the cleavage reaction seems to involve accessory proteins in addition to RNA polymerase. In E. coli, the GREA and GREB proteins stimulate the polymerase to cleave and release the 3' fragment and to resume transcription elongation. Thus, GREA and GREB can be defined as cleavage stimulatory factors. The physiological role of transcript cleavage reaction catalyzed by RNA polymerase has not been determined. However, one accepted hypothesis is that transcript cleavage serves to rescue RNA polymerase that are arrested during elongation. It has been shown that arrested RNA polymerase complexes can be reactivated by transcript cleavage in vitro. The cleavage stimulatory factors GREA and GREB were shown to release the RNA polymerase from the elongation arrest. GREA can suppress the formation of arrested complexes in vitro. It can act only if it is present before the polymerase arrest. GREB can act after the polymerase has stopped. Its action is triggered by the appearance of an arrested complex. It has been suggested that the GREA and GREB stimulate promoter escape as some promoters in vivo. Another role that has been presumed for transcript cleavage is to increase the fidelity of transcription by facilitating the removal of misincorporated nucleotides. To accomplish transcript cleavage and the elongation resumption, the RNA polymerase, after it has stalled, is assumed to either move backwards along the DNA template or undergo structural changes so that the catalytic site is repositioned to the new 3' end to which bases should be added. The ability of the polymerase to withdraw, to cleave and discard the 3' end of the nascent transcript to replace the removed sequence with a newly synthesized one, and to continue with elongation in a template-directed manner, is what makes this process so suitable for proofreading. The old-fashioned view of elongation process is a smooth forward motion during which RNA polymerase moves 1 BP along the DNA template for every base added to the newly synthesized RNA chain. Might still hold for some regions of DNA. However, evidence has accumulated for a different type of movement of the polymerase during elongation. Thus, a new model for RNA polymerase translocation has evolved, the inchworm model, which has been described as the movement of RNA polymerase on a DNA template in discontinuous inchworm-like fashion. The model predicts that the process of RNA chain elongation is a cyclic process that consists of discrete translocation cycles. Each cycle involves a steady compression of the RNA polymerase on the DNA template followed by a sudden expansion. According to this model, the upstream back boundary of the enzyme moves steadily during elongation as the RNA chain is extended. However, the downstream front boundary of the enzyme does not move while several nucleotides are added. It then jumps. That is, it moves 7 to 8 BP along the DNA. At the beginning of each cycle, RNA polymerase stretches across 35 NT of template DNA. It gradually compresses from the back end until it covers only 28 NT. It then releases from the front end and stretches back to cover 35 NT. As the RNA polymerase compresses, the nascent RNA chain model also makes predictions about the existence of more than one RNA binding site on the ternary complex. It has been shown that the nascent RNA interact with at least 3 sites on the E. coli polymerase, 2 on the B subunit and 1 on the B transpose subunit. It is believed that together the DNA and RNA sites account for the remarkable stability and flexibility of ternary complexes. However, the precise size placement and strand specificity of these nucleic acid binding sites are currently being elucidated. Good day, this is Professor Will, CBMD, PhD, today, Saturday, January 11, 2020 at 7:57:51 p.m. Eastern Standard Time. And this concludes my reading on transcriptional elongation. Hope you enjoyed listening. Have a great day.